I mean, the big problem we have is that there's really no cure for HIV. There's no cure for HIV, there's no vaccine for HIV. And for every person that you put on the antiretroviral therapy, 3 or 4 new people become infected. And so, you know, we really can't treat our way out of the HIV epidemic. I mean, maybe if we could get everybody onto antiretroviral therapy, we'd have so many people on antiretroviral, because when you're on antiretroviral therapy, your chances of spreading the virus are much, much, much lower. So if we could get enough people on antiretroviral therapy, then perhaps that would create enough of a bottleneck that the virus couldn't really spread to new people, that we could end the epidemic. That's theoretical, and the cost of getting everybody on there. The big problem is, particularly in places like Africa, there's a lot of people you don't know who are HIV positive. You know, you can't just test everybody. So, you know, what it really comes down to is thinking about ways of doing HIV prevention. And the ways in which you can do prevention, the prevention toolkit is really that. And you have both scientific and biomedical interventions, all the way to sort of behavioral and communication types of interventions. And I would say, I think one of the lessons we learned over the last 2 to 3 years, 3 to 4 years, is that male circumcision is one of the best HIV prevention strategies. And there were 3 really nice trials that were done, one in Kenya, one in Uganda, and one down in South Africa, looking at male circumcision and looking at the spread of HIV through communities where men had been circumcised. And for example, the trial that was carried out in Uganda, using the cohort of individuals that I work with over there, that trial was stopped early when they found that they could decrease the spread of HIV by about 60% in control groups where the men had been circumcised. So this is now a major behavioral intervention, or, you know, it is difficult though to.
The circumcision, does that prevent people from getting it or keeping it, or both?
We think it's both. I mean, circumcision, so it looks like there are cells in the foreskin of the penis which stabilize HIV and either help it, you know, I don't know that much about the biology of male circumcision, but I think could stabilize HIV whether you are passing it on to somebody or whether you are receiving it yourself. But I think it's really in the direction of male to female. So if you're circumcised, the HIV doesn't have those cells that can really sort of stabilize the virus and help shuttle it into, you know, into the person's partner. I mean, actually, there's just a related story. There's my lab works on a protein in semen called SEVI, the semen enhancing virus factors. I can't remember the I stand for. And this is a factor that's present in semen which enhances HIV infection by like 200,000-fold. So if you're, you know, this is one of the reasons why we think the virus really spreads through semen, is because there's this factor actually in the semen itself which really stabilizes the virus and helps move it, you know, on. Because HIV is actually not a very stable virus. The HIV envelope is very labile. It's not easily stabilized, and so HIV has to take advantage of, you know, different host proteins, different factors in our bodies to stabilize it, to help move it on. But anyway, there is a whole toolbox of behavioral interventions, some which work better than others, like microbicides, which are basically creams that women would use and put on their vagina to stop the spread of virus. These haven't been very effective. But certainly there's been different types of education interventions or harm reduction. I mean, things that, you know, working with injecting drug users and getting them clean needles, things like this, which I think are generally accepted as being quite successful. But again, really the big problem with prevention is we just aren't getting to a lot of the people who are at risk. You know, the emphasis has been on treatment, the emphasis has been on getting drugs to people. What we need to do is put greater emphasis on some of these types of preventative measures, and particularly in places like sub-Saharan Africa. So the question that people in my lab and myself and my friends and we talk about a lot is, you know, how do you actually make an HIV vaccine? Why have we failed to make a vaccine, and how can we do it? You know, and I'll say you really got to understand why it's so difficult to stop HIV. You really have to think about the life cycle of the virus and what this virus is doing. You know, HIV is an enveloped retrovirus that binds to a receptor on our CD4 T cells. And so these CD4 T cells right there, and probably most of you know, these CD4 T cells are critical components of our immune system. So HIV, they're critical for producing, CD4 T cells are critical for helping B cells produce antibodies. So right there, HIV is attacking one of the key cell types that we need to produce an effective immune response against the virus. But once the virus envelope binds to these receptors, the viral membrane fuses with our host cell membrane, and that allows this inner viral capsid, it's kind of an egg-like structure, to get into the cytoplasm of our cell, and from there the viral RNA is converted into DNA. This is the process of reverse transcription. And it's really in this process, the reverse transcriptase enzyme, which helps convert the RNA into the DNA. This is a really crappy enzyme, and it introduces a lot of errors and a lot of mistakes into the newly synthesized HIV DNA, which that cDNA then gets integrated into our own host genomic DNA. And then from there, it is expressed, viral proteins are made, and then the virus is assembled and goes on to infect the new cell.
So like you said about T cells and the count of T cells, if this infects T cells, does that mean that it's killing them? And that the number of your T cells is how many are still alive in your body? Is that how it works?
Yeah, I mean, so, yeah. When the virus infects the cell, it replicates for maybe 3 or 4 days in those cells, and then destroys the cell. The process of replicating. I mean, you're making a lot of viral proteins here. In general, your cell does not want a foreign protein produced in it. Makes a lot of proteins. You've got these viruses budding off, so they're really disrupting the integrity of your membrane. So, yeah, the virus will replicate for maybe 3 or 4 days and kill the cells. In a small percentage of the cells, it doesn't kill the cells, it just sits in your genomic DNA. Doesn't produce any viral proteins, doesn't go anywhere, it just lands in there and then stops replicating. I'll talk about that in a second. So it can become sort of a latent virus. But yeah, and when we talk about CD4 T cell counts, like you measure it in a patient, you're really talking about the number of CD4 T cells per milliliter or per microliter that are still circulating in the blood. Now, I will say that one of the things we've learned over the last couple of years is that actually, when you first get infected with HIV, we tend to think about HIV as just being in our blood, but it really isn't. I mean, it infects tonsil cells, but it really infects your gut. It's CD4 T cells that are in your gut, in the gut epithelium, and when the virus infects you, actually, in the first 2 to 3 weeks after infection, you lose about 50% of your T cells in your gut. And so you'll have this period right after you get infected where the virus is replicating. The amount of virus produced in your body is very high. Maybe you get a fever, you feel sick, you get flu-like symptoms. Then your immune system begins to control it, and virus starts to decrease, and then it starts to move to other parts of the body, and then that's when it will begin to infect and destroy circulating T cells that are in your blood. So when we measure viral load, we're only measuring the circulating T cells. It doesn't tell us anything about what HIV is doing in other parts of the body. And individual is different. So you might have somebody that has high CD4 T cell counts in the blood, and we say, okay, this person's doing really well, but perhaps there's other compartments of the body, you know, like tonsil, you know, the gut, you know, or something that it's replicating and you're getting T cell depletion there. So this is something that we're really, it's kind of new to us because for years, I mean, like for many years, we really only measured the virus and how it circulates in the blood, but it turns out it's a much more complex infection.